So our next speaker is Rayna Bell. She grew up in California, and she did an undergrad and a postdoc at UC Berkeley. But in between, she went to Cornell for her PhD. And then she actually went back to the East Coast to be at the Smithsonian, like Washington, D.C., the huge museums in Washington, D.C. She worked there as a curator for a while. And then right before COVID, she moved back to California, and she's now assistant curator of herpetology at the California Academy of Sciences. You know, California is wonderful. Everyone knows this. But I guess not wonderful enough, because Rayna travels to the tropics a lot, too, to find all kinds of funny frogs and understand their ecology and evolution.
Great, thank you. Yes, I was also motivated a lot by my first trip to Gabon, so something that I didn't know that I shared with a very distinguished scholar, so it's nice. Thank you so much for inviting me, Virginie. And so today I'll be talking about a project that has been going on for a few years now, and it's focused on the diversity and evolution of visual systems across the frog tree of life. And so this research is in collaboration with vision biologists and herpetologists who are interested in what the organisms that they study can see. So this is in collaboration with the Fujita Lab at the University of Texas Arlington, the Stryker and Gower Labs at the Natural History Museum London, and then a large team of vision experts, including the two really fantastic postdoctoral researchers that were hired for this project, Kate Thomas and Ryan Schott, and emeritus faculty who are vision experts in their own rights, Ellen Lowe and Ron Douglas. And then, as I mentioned, frog vision enthusiasts from 5 continents all around the world. And so our work has been funded by the US National Science Foundation and the United Kingdom's National Environmental Research Council through a special joint funding program that really aims to support cross-Atlantic collaboration. And so that's been a really fantastic opportunity for us. And so through this project, we've also worked with several wonderful undergraduate, postbachelorate, and master's students on different projects, and I'll be talking about some of their work today. So why would we want to study vision? Well, organisms rely on their senses, touch, hearing, vision, smell, to interpret their surroundings, to do really important tasks like finding food, avoiding predators, finding mates, and to follow their circadian rhythm. And so I think these complex traits are really fascinating to study because they're under very strong selection, but I also think they're really fun to study because they give us a glimpse as to how other organisms experience the world. So it's kind of fun to put yourself into another organism's sphere and think about how they're experiencing the world around them. So frogs, I think, are amazing animals, and hopefully you'll be convinced by the end of today as well, because they've adapted to live in a wide variety of environments, from deserts to tropical rainforests. There are species that spend most of their life underground in burrows, and then there are species that are specialists at navigating the complex environments of the tree canopies, mostly in rainforests. And then we have species that are completely aquatic. Most frogs are nocturnal and are primarily active at night, but we also have some species that are completely diurnal. And so all of these factors are influencing how the frog perceives its environment, and vision is playing a really important role in this. And then, of course, frogs are famous because they have this major transition in their development as they metamorphose from an aquatic tadpole to an adult. And so as you can see in this cartoon here, the total availability of light, as well as the spectrum of available light, diminish in water relative to air. And so the light environment can be quite different over the course of an individual's lifespan. And so because of this complex life history and the diverse habitats that frog inhabit, they're a really exciting group to study how eyes have responded to these different visual pressures. And so today I'm going to give a quick introduction to the main features of the vertebrate eye, in case you are not vision experts, which I was not when I started this project. And then I'll share some of our current research on the diversity and evolution of the visual system. So the vertebrate eye is a very specialized light detection organ that has a few very distinct components. And so here we're looking at the eye in cross-section, and light is going to first enter through the cornea, pass through the pupil, and then be projected from the lens onto the retina. And the retina is a thin layer of neural tissue that will absorb light and then pass the signal to the brain through the optic nerve. And so changes in the size of the eye and these different structures that light is passing through on the way to the retina, so the cornea, the pupil, and the lens, will determine how much light reaches the retina and also whether certain wavelengths of the light spectrum will be filtered out before the retina has the chance to detect them. And so here we're zooming in on the major cell types of the retina, and the ones we'll be talking about today are the photoreceptors, the rods and the cones. And these are the cells that are responsible for absorbing light and producing the signal that will ultimately result in vision. So, as you might infer from their names, rods and cones are typically different sizes and shapes. And they also differ, importantly, in their sensitivity to light. So rods are optimized for dim light vision. They're very sensitive, but they react slowly, and so the images that they produce are less sharp. Cones, on the other hand, are optimized for bright light and color vision, and so they are less sensitive to light, but they react quickly and they can produce sharp images. Rod and cone photoreceptors also vary in their sensitivities to different wavelengths in the light spectrum, and the light spectrum ranges from red to ultraviolet. And so we can use a tool called microspectrophotometry to measure the sensitivity of individual photoreceptor cells to the entire light spectrum and then calculate which wavelengths or which colors they are most sensitive to. And so this absorbance curve here shows the light spectrum of the absorbance of a typical frog rod photoreceptor, which is the green part of the spectrum. So most vertebrates have just one type of dim light or rod photoreceptor, but multiple types of cone photoreceptors. And these different types of cone photoreceptors are sensitive to different parts of the light spectrum. And so comparing information from these different classes is what enables color vision in the organism. Visual pigments are the actual photosensitive molecules that are going to absorb the photon of light and initiate the cellular response that will ultimately result in vision. And so in the photoreceptor cells, like the rod cell shown here, the visual pigments, which are the little blue dots, are located in the discs of the outer segment of the cell. And so if we zoom in on these structures, we can see that the visual pigment is composed of two partners. We have the opsin protein, which is embedded in the membrane of the disc, and then we have a vitamin A molecule, which is called a chromophore, that is associated with this opsin protein. And so when the vitamin A molecule absorbs a photon, it changes its shape, and this pushes against the opsin protein and breaks their bond and initiates the first step in the light detection pathway, which is called the phototransduction cascade. So vertebrate visual opsins emerge through a series of gene duplications in the ancestor of vertebrates. And the duplicated copies have since evolved DNA and amino acid sequence differences, and so they're considered a gene family. In vertebrates, we have 5 different classes. We have the rod opsin, which is expressed in the rod cell for dim light vision. And then we have the cone opsins, which are expressed in cone cells for bright light vision. And these all differ in the wavelengths at which they absorb light, which is demonstrated by the plots across the top. So throughout the evolutionary history of vertebrates, some groups have lost one or more of the opsin genes, and duplications have also occurred in some groups. So because light detection is determined by the interaction of both partners in the visual pigment, the opsin and the vitamin A molecule, changes to either of those partners can affect the overall sensitivity of that visual pigment. So first we'll talk about the chromophore. So there are 2 forms of vitamin A in vertebrate visual pigments, vitamin A1 and vitamin A2. And changing between the 2 chromophore types, if you maintain the exact same opsin, will completely change how that visual pigment will respond to light. So a change from the vitamin A1 to vitamin A2 will shift the sensitivity to redder wavelengths, and it can be quite dramatic. And so most terrestrial vertebrates use the vitamin A1 form, but vertebrates that live in freshwater habitats typically use the A2 form, and that's because freshwater habitats are red light shifted. So if you think of like a murky stream. On the other hand, we can also have a change in the DNA sequence of the opsin gene. And so this will result in a change in the function of the protein and how it's interacting with that vitamin A molecule. And so we can have changes both in the overall sensitivity of how sensitive that visual pigment will be to a light signal, but also the specific wavelengths that the protein and chromophore partner will be sensitive to. And so the changes can affect the sensitivity to be more towards the red part of the spectrum, but also towards bluer wavelengths of light. And then the final piece of background information that's important to know. So I mentioned that most vertebrates have just one type of rod photoreceptor for dim light vision, but frogs and some salamanders are special because they have a second type of rod that is confusingly called the green rod because it looked green under the microscope when it was first discovered, but it's actually sensitive to the blue part of the light spectrum. And so you can see its sensitivity here compared to the regular rod, which even more confusingly is called the red rod, even though it is sensitive to the green part of the light spectrum. But so what are the benefits of having these two different types of rods or dim light photoreceptors? Well, some behavioral studies in frogs are suggesting that having these two different classes of rods could allow for color vision in dim light conditions, during which cones are not functioning because they're not sensitive enough to detect light in these dim light conditions. And so having two classes of rods could provide better nocturnal vision in frogs and even color nocturnal vision in frogs that would help them navigate at night and identify their mates. And so these red-eyed tree frogs are one nocturnal species in which there's growing evidence that visual signals and color differences between populations are an important part of mate choice, and all of their behaviors are being done under moonlight conditions, during which their cone photoreceptors are not active. And so nocturnal color vision in frogs is a very exciting area of research if you're interested in the behavior and ecology of frogs. Okay, so hopefully now you have an idea of how integrative vision biology is. In this collaborative grant, we are focusing on a few main types of data. We're looking at variation in the photopigments and spectral sensitivity of the photoreceptor cells, the molecular evolution of vision genes, and also the diversity of eye morphology across a broad suite of frogs so that we can get an overview of the diversity of visual system evolution across the frog tree of life and how this correlates with ecology and life history of frogs. Okay, so now I'll share some of our current research in this area. So as I mentioned, the size of the eye and the structures that light is passing through on its way to the retina, and then we'll determine how much light is going to reach the retina and whether certain wavelengths of light might be filtered out before they reach the retina. And then there are properties of the actual visual pigments in the photoreceptor cells that are going to determine which wavelengths of light the animal is sensitive to. And so these are some of the first features that we've set out to characterize across this broad phylogenetic and ecological diversity of frogs. So across vertebrates, larger eyes typically provide better sensitivity and higher resolution or acuity for the animal. So animals that rely heavily on vision to sense their environments typically have very large eyes. Eyes are very metabolically expensive tissues, however, and so the relative size of the eye is expected to vary depending on the ecology and visual needs of a given species. So if they rely heavily on vision, they will invest in eye tissue, and if they don't rely heavily on vision, they will divest in eye tissue. We generated a data set with 220 species, including all anurin families, so that we could characterize how eye size varies across diverse species and ecologies. And fortunately for this work, we were able to use specimens in natural history collections so that we could have very comprehensive sampling. We found overall that species occupying aquatic and fossorial habitats, or burrowing species, have significantly smaller eyes than those in terrestrial or scansorial climbing habitats. And in particular, we found that tree frogs have disproportionately large eyes, which is probably not a surprise if you've looked at a tree frog before. And we think this is because they're primarily active in dim light, so they need to maximize their ability to detect light. And they also need to maintain high visual acuity, or the sharpness of their images, so they can complete complex tasks that require precise vision. So if you think about tree frogs jumping from one branch to the next in very dim light conditions. In many animals, including in our own eyes, the lens is pigmented so that it blocks the UV part of the light spectrum. And this is thought to be beneficial because it protects the eye from UV damage. And this blocking of the UV wavelengths can also increase your visual acuity, or the crispness of your images, by reducing chromatic aberration or distortion and scatter. And that's because the UV part of the light spectrum, it has a lot of noise in it. And so UV filtering lenses are typically found in vertebrates with behaviors that are associated with high acuity. On the other hand, there are some animals that use visual signals in the UV part of the spectrum. And so these species have UV transparent lenses so that the UV can reach the retina and be detected by the photoreceptors. And so understanding whether the lens is blocking UV or transmissive to UV is a really important aspect of characterizing the visual ecology of a species. So we set out to measure lens transmission in as many species of frogs as possible. And to measure lens transmission, you need a freshly dissected lens. And so we spent a few years working with collaborators all around the world to collect this data set. And thanks to this extensive collaboration, our sampling includes a strong representation of the ecological diversity of frogs. Overall, our results indicate that both diurnality and climbing behaviors, so again, these tree frogs, are correlated with reduced UV transmission in the lens. And we found that many species, primarily nocturnal species, have UV transparent lenses, suggesting that they are somewhat sensitive to this part of the light spectrum, and that UV light could play an important role in their ecology and behavior. Lens shape is another important aspect of the visual system. So it's known to differ between aquatic and terrestrial vertebrates to facilitate vision in different optical media. And this is because of the way that light behaves when it's moving between air and water. And the inside of the eye is basically similar refractive properties to water. And so species that are viewing through water have more spherical lenses, and those that are viewing through air have somewhat flattened lenses. And so this presents a really interesting challenge for frogs, which often begin their life cycle as an aquatic larvae and then transition to life on land through metamorphosis. So we used CT scans of museum specimens to quantify lens size and shape across a broad diversity of frogs. And this sampling also include matched tadpoles and adults of the same species for many of the species that we looked at. And so we found that yes, tadpole and adult lens shapes differ. Tadpoles typically have rounder lenses and adults typically have flatter lenses, except if the adults are fossorial or aquatic. They retain round lenses, or they have lenses that look more similar to tadpoles. And we expected this result for aquatic frogs, but it was somewhat unexpected for fossorial frogs because they're viewing through water, not through land. And this spherical lens shape should make them myopic or nearsighted. But this could be adaptive because they're likely focusing on objects at short distances in their underground burrows and also in the brief amount of times that they're above ground. Okay, and then we have the diversity and the spectral sensitivities of the photoreceptors themselves, which of course have important implications for the visual abilities of a given species in both bright light and dim light environments. So for example, vertebrates that are primarily active at night tend to have a rod-dominated retina, whereas those that are active during the day will have high densities of cones. And species that have reduced reliance on vision, like burrowing mammals, have lost entire classes of photoreceptors. On the other hand, species that communicate in the UV portion of the spectrum have tuned their typically violet-sensitive cones to be sensitive to even shorter wavelengths into the ultraviolet part of the spectrum. And so we set out to quantify the diversity of photoreceptor sensitivity across the frog phylogeny to understand what cell types are present and what wavelengths they're sensitive to. And again, we spent a few years working with collaborators around the world to gather this data set. And for a very small subset of species, we also have this paired tadpole adult sampling. So we found that most frogs have rod-dominated retinas, which is expected due to their largely nocturnal lifestyles. But species with diurnal activity periods have abundant and very well-developed cones. And then we also found substantial variation in the spectral sensitivity for each different photoreceptor class among different species. And so this suggests that species are tuning their visual systems to maximize peak sensitivity to different parts of the light spectrum, which is presumably associated with the different environments that they're active in, and also different colors that are maybe relevant for them to be able to see well based on their behavior and ecology. So through all of these morphological and optical data sets, we're seeing some clear trends in the ways that frog visual systems are associated with their lifestyles, and in particular, that scansorial frogs, or tree frogs, seem to have really excellent visual systems, whereas fully aquatic species and burrowing species have reduced eyes that are similar in a lot of ways to the eyes of tadpoles. And so what is the genetic basis of all of this variation? Well, for all of the species that we've collected the lens transmission and photoreceptor sensitivity data for, we also have whole eye gene expression data, and so this gives us the opportunity to look at the molecular evolution of a broad suite of vision-related genes and to match them with our phenotypic data, like the photoreceptor sensitivity measurements. And so I'm going to share the results of 2 more detailed projects in which we've applied these approaches. So, as I mentioned, the aquatic and terrestrial environments differ both in the intensity and in the spectral composition of light. And so the visual system of frogs that have an aquatic larval stage and then transition to life on land are encountering vastly different light environments within their lifespan. And so in this study, we were interested in how whole eye gene expression might differ between these life stages. And for this comparison, we focused on a species that is common in the US, the southern leopard frog, and we preserved whole eyes and RNA later from 6 wild-caught tadpoles and 6 wild-caught juveniles for RNA-seq. And so broadly, the questions that we're interested in are which visual genes are differentially expressed between life stages, and also whether the spectral sensitivity of the visual system differs between the larval and adult life stages. So in other words, how is the visual system changing in the aquatic versus the terrestrial light environment? So we found that over 42% of genes were differentially expressed in the eyes of tadpoles versus juveniles, including a wide suite of genes related to visual function, like visual opsins, cone-specific and rod-specific phototransduction genes, and also lens crystallins. And I'm going to zoom in on one of these genes that we were particularly excited about. So we found significant upregulation of this gene, CYP27C1, in tadpoles. And this gene has been implicated in mediating the conversion between the A1 and the A2 form of the chromophore, so the key component of the visual pigment. And so this shift should result in different photoreceptor sensitivities while maintaining the exact same opsin sequence. And so indeed, when we measure the sensitivity of the tadpole and adult photoreceptors with microspectrophotometry, we found red-shifted absorbance of visual pigments in the tadpoles, which is consistent with them using the A2 form of the chromophore. And shifts to these redder wavelengths are common in vertebrates that are inhabiting freshwater environments because the light environment of freshwater is red-shifted. So this gene, CYP27C1, is just one of the many suites of visual genes that were differentially expressed. But overall, this set of results indicates that there's decoupling of gene expression between the life stages that enables frogs to adapt their vision to distinct aquatic and terrestrial light environments over the course of their life cycle. So based on our microspectrophotometry data of the photoreceptor cells across all frogs, we know that there's variation in the spectral sensitivity of the rods and cones among species. And there are also species in which we didn't detect a particular cell type with the microspectrophotometry, suggesting that it could be absent, although it's not a super quantitative approach, so we can't be sure. But we can pair these data with our cross-species RNA-seq data to characterize gene loss and duplication, and also how sequence evolution might be associated with the spectral differences in the photoreceptors among different species. So we sequenced eye transcriptomes from 82 species, and we obtained additional sequences from genomes to produce a data set of 121 species spanning 34 families of frogs. So first, we assessed which visual opsins were retrieved from the transcriptomes and genomes, and then we used PAML to test for differences in selection, so the DN-DS ratio, on these genes using typical codon models in PAML, and also models that partition branches based on differences in species ecology. So these clade models test for evidence of a codon-site class that demonstrates a shift between pre-partitioned or user-partitioned groups in the foreground and in the background. And this can be any combination of clades or branches within the phylogeny. So in the cartoon example here, we can partition the tree with respect to diurnal versus nocturnal activity periods, for example, to test if a partitioned model is a better fit than the null. And so if the test supports differences in selection between the two groups, this could indicate functional evolution and adaptation. In cases where the PAML analyses found significantly elevated DNDS ratios in a particular group, so for example, if we saw elevated DNDS in diurnal frogs compared to non-diurnal frogs, we used a method called RELAX that assesses whether this elevated DNDS ratio is a product of relaxed selective constraint or a lack of selection against change, or if it's due to adaptive selection or selection for change. So across our data set, we consistently recovered 4 visual opsins for each species with some notable exceptions. The 4 dart frogs in our data set were all missing the cone opsin SWS2, and the 2 arthroleptids, which are an African frog family, were both missing the cone opsin SWS1. For the dart frogs, there are full genomes for 2 of the species, and so it's more reliable to confirm the absence of this opsin. And we also didn't find any blue-sensitive rods in our MSP analyses. Unfortunately, for the arthroleptids, there are no genome sequences for this family at this time, and so this will require much greater sampling and hopefully a genome sequence to really confirm the loss of SWS1 in this group. And then finally, we found only 1 case of opsin duplication in our data set, the African bullfrog, which has distinct copies of the LWS opsin on the sex chromosomes and the possibility of sex-specific visual adaptation. And unfortunately, our MSP data, or microspectrophotometry data, is not for a female in this species, and so we don't know if the 2 different copies have different spectral sensitivities, but the sequences are quite divergent, with only 93% amino acid identity. So overall, we found similar levels of selective constraint among the opsins, and using a few different methods for site-specific positive selection, we found statistically significant positive selection at a small proportion of sites in 3 of the 4 visual opsins, which is the graph shown on the right. And then, I'm running out of time, so I'll hurry, but when we were looking at the tests for selective constraint with respect to the different ecological partitions, so again, in these models, species with contrasting ecologies can have different evolutionary rates. This is the suite of different traits that we're comparing across species, so diurnal versus nocturnal, different habitats that they live in, geographic distribution, because this affects the photoperiod they're exposed to, and life history. And life history is interesting because some frogs don't have a tadpole stage. They hatch as a miniature frog from an egg directly, and so they're not spending any part of the life cycle in water. So for each of the genes, we found a significant result with at least 1 of the ecological partitions. And in the cases where we investigated these further with that RELAX method, there was strong support for these being as a result of relaxation of selective constraint, as opposed to positive selection for change. And so the final piece is that we're now comparing the visual opsins and the known spectral tuning sites in other vertebrates, so the known amino acid substitutions that cause changes in the spectral sensitivity, either to redder wavelengths or bluer wavelengths, with the variation in our data set and the observed changes in sensitivity from our microspectrophotometry data sets from those same animals. And so this is the lightning summary of what is most important to know, which is that frog eyes vary in many different ways to enhance their vision in the different habitats that they live in. They have 2 classes of rods, which might provide the ability for nocturnal color vision, and that the life cycle of frogs varies, and this puts different demands on the development and function of their visual systems. So if there's time, I'll take a question.